Welcome to Medicinal Chemistry 2 lectures. Hi everyone, this is Dr. Poorna Nagashri explaining about Medicinal Chemistry 2 lectures under B Pharmacy. So in this, I am going to deal with anti-anginals, vasodilators under that, and in vasodilators, I am going to deal with the mechanism of action of nitrovasodilators. Before going to the nitrovasodilators, let us see the classification of anti-anginals. So the vasodilators are basically divided into three different types because vasodilators are those drugs which are used to dilate the vessels. The blood vessels are like it could be arteries or it could be veins. Hence they are classified as arterial vasodilators, venous vasodilators and arterial and venous vasodilators. These are also called as mixed vasodilators. Now under arterial vasodilators, we have drugs like hydralazine, minoxidil and diazoxide. Under venous vasodilators, which we are going to discuss in detail about their mechanism of action, these are nitrovasodilators. Under arterial and venous vasodilators, which are also called as mixed vasodilators, sodium nitroprusside, it is the drug. So many of these vasodilators are also used as antihypertensives. So coming to the venous vasodilators, these venous vasodilators are further divided into two types, that is nitrites and nitrates. So these are called as nitrovasodilators. Next, last we also have miscellaneous drug. This miscellaneous drug is called as dipyridamole. This is also used as an antiplatelet drug. Now under the nitrites and nitrates, the drugs to be classified are amyl nitrite, nitroglycerin, which is also called as glyceryl trinitrate, pentaerythritol tetranitrate and isosorbide dinitrate. So now I am going to discuss about the mechanism of action of these nitrites and nitrates. Nitrites are usually written as CONO or CONO or CNO2. So, and these nitrates are commonly called as nitrates, CNO3 forms. So, these nitrates and nitrates are CNO2 and CNO3 forms, and hence they are called as nitrovasodilators. Let us go in detail about the mechanism of action. Before going to the mechanism of action, it is mandatory to know about the contraction and relaxation, how it happens in case of skeletal muscles as well as in the smooth muscles. So, whenever there is a contraction, we all know that calcium is involved in that contraction, and of course in the relaxation also it is involved. So, how exactly the calcium is involved in causing the contraction of a muscle? So, let us see here. The dotted one is the cell membrane. So, this is the extracellular region and this is the intracellular region. So, whenever calcium enters through its calcium channels, that are ion channels. So, whenever calcium enters through these ion channels inside the membrane, there the free calcium forms a complex with calcium and calmodulin. Calmodulin is a protein with which the calcium binds and forms a complex. So now this calcium calmodulin complex plays a very important role in causing the contraction. MLCK, this is nothing but myosin light chain kinase. MLCK is myosin light chain kinase. This is an enzyme. So let us see what is its importance. We know that along with the calcium for the contraction, actin and myosin are very much required. Now, the main constitution of this myosin is of heavy chains as well as light chains, and each myosin unit will be having two heavy chains, and further each of this heavy chain unit will be paired with two light chains. So, myosin is comprised of heavy chains as well as the light chains, and heavy chains are again further they are constituted by two pairs of the light chains each. So, heavy chains have heavy and light chains are lighter in their constitution with respect to the molecular weight. Now, this myosin forms a complex with the actin and then causes the contraction. So, in this the light chains are going to act as the substitute for the contraction. So, let us see how they are acting as the substrate for the contraction. So, the MLCK, myosin light chain kinase is an enzyme which is helping in the contraction process. The myosin light chain kinase is converted to myosin light chain kinase activated form. The MLCK star is activated form. So, as the MLCK is converted to the MLCK activated form, this happens in presence of calcium calmodulin complex only. So, without the calcium calmodulin complex, there is no activation of the myosin light chain kinase. So, now this is the second important step in the contraction. So, the activated form of the myosin light chain kinase again helps in the conversion of this myosin light chain. The LC stands for light chain. This myosin light chain is getting phosphorylated. The myosin light chain which is phosphorylated, it combines with the actin and causes contraction. So, let us see in detail about the contraction again. The calcium which is entering through the ion channels, that is calcium channels of the membrane, enters inside the membrane and forms a calcium calmodulin complex. So, this is the crucial step number 1. Next, in presence of this calcium calmodulin complex, the myosin light chain kinase, which is abbreviated as MLCK, is getting converted to the activated form, that is MLCK star. So, this is the second most crucial step in contraction. Now, in presence of this activated MLCK, the myosin light chain is getting phosphorylated. So, this is the third crucial part in the contraction. Then the final step is the myosin light chain which is in the phosphorylated form combines with the actin and causes contraction. Okay. So, here calcium calmodulin complex formation, MLCK activation and myosin light chain phosphorylation. These three are the important steps to cause any skeletal muscle contraction or smooth muscle contraction. Now, how the relaxation happens? The relaxation happens in a simple one step in presence of enzymes called as phosphatases. The myosin light chain which is phosphorylated will be dephosphorylated. So, once the dephosphorylated myosin light chain is present, this automatically causes the reverse of contraction, that is relaxation. Now, in this contraction process, there are two particular key modulators which are called as cyclic AMP, CAM and cyclic GMP. So, the cyclic AMP and cyclic GMP, they mainly help in the vasodilation process. So, let us see one by one how the CAM and CGMP are helping in the relaxation process. So, if I have to explain the cyclic AMP role with the help of a drug, so let us take any beta agonist drug, and this beta agonist drug, once it passes through the membrane, through the receptors, There, in presence of adenylate cyclase, that is AC, the ATP is converted to cyclic AMP. Now this cyclic AMP is responsible for the conversion of MLCK to MLCK phosphorylated form. Okay. Please remember here I am talking about the MLCK phosphorylation but not about the myosin light chain phosphorylation. So if the cyclic AMP is helping in the conversion of MLCK to MLCK phosphorylated form, then the activated form of the MLCK will be much reduced. So as the MLCK is getting converted more and more to MLCK phosphorylated form, and due to the availability of the cyclic AMP, the MLCK phosphorylated form concentration is increasing. And here there will be unavailability of the MLCK activated form. Hence, the MLCK activated form is not available to phosphorylate the myosin light chain. There will not be any contraction. So, in this way, cyclic AMP is helping in the vasodilation here. So, this is the role of the cyclic AMP in vasodilation. Hope you all understood this one. So, there are 3 steps to remember in contraction and relaxation. That is, after the entry of the calcium, the formation of the calcium calmodulin is first step. Then MLCK activation is the second step. Third is the phosphorylation of the myosin light chain. The cyclic AMP, what it is doing in this contraction relaxation process is the MLCK is not getting converted into its activated form. So, because the cyclic AMP is converting MLCK into its phosphorylated form, and hence MLCK activated form is not available for further contraction. Hope this is clear to all of you. Next, we will move on to the actual mechanism of action of the nitrovasodilators. Once we understand this calcium calmodulin complex and the role of the CAMP, then we can easily understand the role of the CGMP through these nitrovasodilators. In this picture, I have given 2 drugs. One is organic nitrates that I am talking about, and the second is sodium nitroprusside, which is a mixed vasodilator. So both these drugs, they act through the same mechanism. Organic nitrates, first what they will do is they get converted to the nitrites. So nitrates are converted to nitrites. Now these nitrates are converted to the unstable nitrosothiol form. Once the nitrosothiol form is obtained, from here the key step, that is the release of the nitric oxide, NO. This is a free radical and which helps in the activation of the guanylate cyclase, that is GC. So once the released nitric oxide is activating the guanylate cyclase, in presence of the activated guanylate cyclase, the GTP, guanosine triphosphate, is converted to cyclic GMP, cyclic guanosine monophosphate, and this cyclic GMP is going to cause the relaxation. So now there are 3 key important steps in this nitrovasodilators mechanism of action, that is the release of the nitric oxide from the organic nitrates or nitrites. That is the first step. The second step is activation of the guanylate cyclase. That is the second step. Third one is the formation or increase in the concentration of cyclic GMP. So because of all these 3 steps, there is relaxation. Now the sodium nitroprusside is also acting via the same mechanism, where this is also releasing the same nitric oxide as a free radical, and this nitric oxide which is present inside the smooth muscle causes the same type of relaxation just like the organic nitrates and nitrites, right? So this is the mechanism of action of nitrovasodilators, where the organic nitrates are converted to nitrites, further they are converted to the unstable nitrosothiols, and then to the free radical which is very reactive, that is nitric oxide. This nitric oxide helps in the conversion of the guanylate cyclase to the activated form. The activated guanylate cyclase helps in the conversion of the GTP to cyclic GMP. So as the concentration of the cyclic GMP increases, just like in case of the cyclic KMP, there will be more relaxation, because even this cyclic GMP is also going to convert the MLCK into MLCK phosphorylated form. So because of this, the cyclic GMP and cyclic KMP both are actively involved in the vasodilation process. So now let us see through another last picture how these nitrovasodilators are working. Before going to the last picture, I just want to show you the theoretical slide where I have just formulated into 3 steps about the mechanism of action of nitrovasodilators. So these drugs are rapidly denitrated enzymatically in the smooth muscle cells to release the active free radical nitric oxide, which is a reactive one. Next, the nitrates, that is RNO3, are converted to RNO2, that is nitrates, and further they are converted to nitrosothiols, that is RSNO. And these nitrosothiols are converted to the nitric oxide. So the release of the nitric oxide helps in activating the CGMP or CAMP, and this leads to the phosphorylation of the MLCK. The MLCK phosphorylated form causes the relaxation. Now this phosphorylated MLCK fails to cause the contraction, and this relaxation could also be another factor due to the reduction in the calcium entry. We have seen in the previous slides that the MLCK phosphorylation is reducing the activated MLCK form, and hence there is no myosin light chain phosphorylation, and hence actin cannot combine with the myosin light chain, and hence no contraction. There is another thing, that is, maybe in the first step itself, if there is a reduced calcium entry, so this reduced calcium entry could lead to the decrease in the contraction. So in spite of the phosphorylation and blocking the MLCK activated form, we can also stop the calcium entry in the entry level itself. So even that can also lead to the relaxation, as there is no availability of the calcium calmodulin complex formation. So this is the last slide of the nitrovasodilators mechanism of action. Where whatever that I have explained in the previous slides was shown here. So these are the nitrovasodilators which are generating the nitric oxide, the free radical, through nitrate to nitrite, nitrite to nitrosothiol. Then from nitric oxide, the guanylate cyclase is getting converted to guanylate cyclase activated form. Now this activated form of the guanylate cyclase helps in the conversion of the guanosin triphosphate to cyclic guanosin monophosphate, GTP to cyclic GMP, just like the ATP to cyclic AMP. Now this cyclic GMP helps in the conversion of the myosin light chain to myosin light chain phosphate form, and this causes relaxation. So because of the conversion of this MLCK to MLCK phosphorylated form, there will be decrease in the concentration of MLCK activated form, and hence contraction is getting blocked. Right? Understood, friends? So now the endothelial cells, they release EDRF, that is endothelium derived releasing factors. Even this is also having the same vasodilator effect just like the nitric oxide. So this EDRF and NO, they act in the similar manner, and hence this nitric oxide is acting like a vasodilator. It is promoting the series of events which can help in the relaxation or vasodilation. So this is the mechanism of action of nitrovasodilators, which we can easily understand in 3 simple steps. That is by the formation of calcium calmodulin complex, then myosin light chain kinase activation, then myosin light chain phosphorylation, then contraction due to the binding of actin with the myosin light chain. So this is in brief about the nitrovasodilators. Once this contraction happens, the nitrovasodilators, they are going to release the nitric oxide, and this nitric oxide again is going to activate the guanylate cyclase, and the activated guanylate cyclase is going to increase the concentration of the cyclic GMP, and the increased concentration of the cyclic GMP is going to convert the MLCK into MLCK phosphorylated form. So finally, because of the formation of the MLCK phosphorylated form, there is relaxation. Thank you one and all for watching this.